So today's lesson we're going to talk about the light reactions of photosynthesis. So here I have my thylakoid membrane. This is the stroma outside of the thylakoid, and all of this is happening inside of the chloroplast. Okay, so these are the little thylakoid discs that are inside the chloroplast. So we have photosystem 2, which happens to come first. It just happened to be named second. It was identified second. Photosystem 1, we have PQ, cytochrome complex, PC, FD, NADP plus reductase is an enzyme, and the ATP synthase. So we're going to walk through this. So the first thing that we need for photosynthesis is some light. So light comes in, and it excites the electrons inside photosystem 2. And it causes some electrons to leave photosystem 2 and go to a primary electron acceptor that's going to carry them to PQ. It's going to carry them to the cytochrome complex, which is going to cause this to open. This cytochrome complex channel is now going to open, and then the electrons are going to jump to PC, and that's the first step of our process here. So the light excited the electrons inside photosystem 2. A primary electron acceptor carried those electrons to PQ, to open the cytochrome complex, and then to PC. The other ingredient that we need for photosynthesis, for the light reactions, is water. So what happens is a water molecule gets split into hydrogen ions, electrons, and some oxygen gas. I know that equation is not balanced. I'm not going to worry about balancing at this point in time. We just really need the 3 elements from the water splitting. So what happens is the oxygen is not needed for the plant. It's a waste product, so it's going to exit the chloroplast, and it's either going to exit the plant through the stomata because it doesn't need any, or it's going to go to the mitochondria for cellular respiration, which we'll talk about in another section. What's really important is these hydrogen ions and these electrons. So first we're going to talk about the hydrogen ions. And when these electrons jump through the cytochrome complex and open it up, it's actually so that these hydrogen ions can go through the cytochrome complex and start building up inside the thylakoid space. So every single time these electrons jump through the cytochrome complex, more hydrogen ions are going to fill in the space here inside the thylakoid. Okay, so the purpose of the water, one part of it, is to provide hydrogen ions because we're going to build a concentration gradient inside here. All right. We're going to get to the electrons in a minute, but for now, we have the light, electrons jump, channel opens, the cytochrome complex opens as a result of those electrons jumping through, and we start building up hydrogens. Now we have an empty space here. We lost some electrons, and this is where this particular electron is going to go. It's going to eventually fill in the empty space caused by the light. Okay, so this one is all set. So that's photosystem 2. Now we have this hydrogen ion gradient here, and we're going to use that gradient to cause the ATP synthase to take an ADP molecule and phosphorylize it should have been an arrow, to make ATP. which is our energy, right? Cellular energy. And what happens is we have a concentration gradient. All these hydrogen ions start rushing through the ATP synthase, causing it to turn, and it adds the phosphate to the ADP to make ATP, and that ATP will go to the Calvin cycle for the second part of photosynthesis. The next part is photosystem 1. So the same thing happens. We have light that hits the chlorophyll inside that photosystem. It excites the P700 photosystem, and it causes an electron to leave, and it jumps to this FD molecule here in our membrane, and it jumps down to the NADP plus reductase, and the NADP plus reductase is going to take NADP plus, and it's going to add electrons to it, and it's going to add hydrogen ions to make NADPH. The NADPH is an electron carrier, and it's also going to go to the Calvin cycle. So we will see the NADPH and the ATP again. So we have our electrons that left here, right? They left photosystem 1, and they're going to get filled in by the electrons that ultimately left photosystem 2. Those electrons are going to fill in that gap. So we call these electron transport chains. So where this system here, where the electron jumps from here, from the P680 to the PQ, to the cytochrome, to the PC, that whole thing right there is called an electron transport chain, or an ETC for short. And then over here, we have another electron transport chain involving the NADP plus reductase. So we have a second electron transport chain within photosynthesis light reactions. So just to recap, light excites the electrons in our two photosystems. It causes electrons to jump through electron transport chains. The electron transport chain for photosystem 2 involving cytochrome, the cytochrome complex, takes hydrogen ions that ultimately came from the water, causes them to go through that channel and build up a hydrogen ion gradient. Those hydrogen ions will flood through the ATP synthase, causing the phosphorylation of an ADP into ATP, right? Our molecule of cellular energy. The electron transport chain for photosystem 1 activates this enzyme, NADP plus reductase, which takes an electron and a hydrogen ion, adds it to NADP plus, which came from the Calvin cycle, which you'll see that in the next part of the process, and it makes NADPH, which is an electron carrier that is going to take those hydrogen ions and those electrons and bring them to the Calvin cycle. And using the energy from the ATP, we're going to take those hydrogens and combine it with carbon dioxide in the Calvin cycle and create our sugar molecule.